My name is Koby Nahmias. I'm the director of the Grass Center for Bioengineering at the Hebrew University of Jerusalem. And today, I'm going to talk about animal-free drug development and everything we did these last 2 years to solve the COVID-19 pandemic. As you all know, drug development is a very expensive endeavor. It costs more than $2 billion in more than 12 years to bring a single drug to the market. The main problem is that most of our research is still dependent on rats and mice, small animals that are very, very different from humans. Rats and mice have different physiology, different metabolism, and different genetics than humans. And a lot of the things that we find in them are simply not relevant to the human condition. To change that, we have invested the last 6 years at the Hebrew University to develop a novel human-on-chip platform. We are taking human cells with human genetics and making small organs that mimic human physiology. The organs that we have include a heart that has different chambers and a pacemaker, lungs, kidneys, liver, and brain, all living together. What makes the platform especially unique is not that we have all of those organoids that are very, very complex and are exactly like us, but the fact that these are bionic organs. We have sensors physically integrated into those organs. Sensors that can measure mechanical activity, electrical activity, and metabolic function in real time. They tell us things that we couldn't even imagine that we could get from animal studies. We can understand basic mechanisms of disease in a way that was not thought to be possible before. The real challenge we faced 2 years ago in trying to understand the novel coronavirus made us very, very unique in this space. You see, coronaviruses are known for many years. Actually, they're thought to evolve here in the Middle East thousands of years ago. Most cases, it gives you a common cold. You cough a little bit, you drink chicken soup, and you go back home. But this new generation of beta coronaviruses like SARS, MERS, or the SARS-CoV-2, which is the novel coronavirus, actually cause severe acute respiratory distress. Your lungs become inflamed, and you get a cytokine storm that damages other organs in your body. What's really unique is that the comorbidities, the problems that will get you to the hospital, are not inflammatory diseases of the lung. It's not asthma. It's actually diabetes, obesity, dyslipidemia, so high levels of cholesterol in your blood. These will get you to the hospital and the ER and maybe even the ICU. So we struggle to understand why this is the case. We partnered 2 years ago with the Mount Sinai Hospital to use our lung model to look at the metabolic changes induced by the virus. And what we saw was incredibly surprising. This novel coronavirus caused this massive accumulation of lipids in the lungs. The virus needed those fats. It actually had to have them to replicate. And we could stop that using Fenofibrate, a drug that was approved by the FDA back in 1975. We could show that lipids could completely disappear, and the virus replication was gone by 4 days of treatment. We took this discovery to the clinic, treating severe COVID-19 patients with funding by Abbott. Those patients were completely transformed. Inflammation of the lungs disappeared in 48 hours. What's more exciting is that they didn't develop a cytokine storm. The rest of the organs were fine. Respiratory support was not needed anymore. We could remove all of these patients from respiratory support in less than a week. And we could release them from the hospital just as fast. The most exciting piece of data came in a month and 3-month follow-up. Usually, COVID-19 causes severe problems down the line, even after you recover from the virus. cardiovascular problems, lung problems, neurological problems. Only one patient reported problems in our study after a month, and actually none of them reported studies after 3 months. That's compared to more than 35% of the population. We're very excited to take this data forward, and we're very thankful for all of the support of the Board of Governors, specifically for the Sam and Rina Frankel Foundation, as well as the Nikoa Foundation that supported my lab all these years. Thank you very much.